In this video, we are going to discuss the phase known as glycolysis, the first step in carbohydrate catabolism. In my course, we're studying two types of cells. We're studying prokaryotic cells and eukaryotic cells. So one of the first things that you need to look at is where is this particular event taking place in each type of cell? Now, because this is a medical focused microbiology, we're studying mainly the bacteria as far as the prokaryotes are concerned, because more of them cause disease. And then we're studying a variety of eukaryotes. So look to the bacteria and the eukaryotes, and when we look at them, we find that both organisms perform this process in the cytoplasm of their cells. So location will be important for your exam. We are going to break this glycolysis down into several phases. It's easiest taught. We talk about it in three phases. The first one being the energy capture phase. This is where we're going to invest some energy into the glucose in the form of ATP to weaken it so that in the second phase, we can break the glucose, which is a 6-carbon molecule, down into two 3-carbon molecules. Now, one of the goals of glycolysis is to capture energy in the form of ATP, and that's not going to be done in either of these first two steps. The last phase is where we capture the energy that we're looking for. So these are the three phases that we're going to walk through. Let's clear the board. And please, any chemical names I write, those are going to be important for the test. If I don't write it, then that's not important at this level, at this step in learning glycolysis. Now, if you progress on, there's a chance that you're going to add to what I'm giving you in this course. You'll get more detailed with each course that you move through. All right, so in glycolysis, we're going to start with a 6-carbon sugar. Let's get the rest of this expo off. So 6-carbon sugar. So this is C6H12O6. Now I want you to bear in mind, one of the topics that was discussed at the first of chapter 5 is oxidation reduction reactions. And in oxidation reduction reactions, we can remove electrons from a molecule, and as we do that, we're oxidizing it. So one of the things we're going to start to do is we want to oxidize this glucose to get the energy out of it. Now in biology, it turns out easy to follow most oxidation reduction reactions. We follow the hydrogens. So one of the goals that we're doing during this carbohydrate catabolism is we're going to try to completely remove all 12 hydrogen from this glucose molecule. So anytime we pull a hydrogen off and we pass it to an electron carrier, you need to keep up with that. Basically, I like to equivalent those reduced electron carriers to little paychecks. I've never had a student tell me that if I brought my checkbook to every class and wrote $2 and $3 checks, that they would turn those down. They said they would hold on to them and go cash them in. Well, the cell uses these reduced electron carriers in the electron transport chain to produce additional ATP. So as we move through, you're looking not only for this ATP that we're supposed to be producing, but also reduced electron carriers. And there's two reduced electron carriers that we focus on. We have nicotine adenine dinucleotide. We're going to abbreviate to NAD. And it will get reduced to NADH. We also have flavin adenine dinucleotide, which will get reduced during these processes to FADH. So please keep up with those as you see those appear as we go through these different phases. All right, so we're starting with a 6-carbon molecule. It's called glucose, C6H12O6. And in the first phase, the energy investment phase, what we want to do is spin some ATP. So I'm representing basically 3 steps of glycolysis here. We're going to do them all at one time. We're going to bring in, ultimately in these 3 steps, 2 molecules of ATP. We're going to remove the phosphate group from each of those ATP molecules, releasing 2 molecules of ADP, and those phosphates are going to be moved and placed onto this glucose molecule. So what we are going to form is a molecule called fructose 1,6-bisphosphate. Now, I want you to stop, take a look at this molecule for just a moment. When you're looking at this molecule, look at these charges. These are 2 like charges. Like charges repel one another. Now, these like charges repelling one another, they're causing this middle bond to become weak. We've done this for a reason. A glucose molecule itself is an extremely stable molecule. We're not going to be able to go in with an enzyme and simply split that thing into 2-3 carbons and move on. We are investing this energy so that we are purposely making this middle bond between these 2 carbons a weaker bond, so that the next enzyme can take this molecule, this fructose 1,6-bisphosphate, and lyse it into 2 different 3-carbon molecules. So the next phase, we end up with lysis. And the lysis will yield 2 3-carbon molecules that are phosphorylated. Now, these are different molecules. If you need to see all of the carbons and the hydrogens that are attached, you can look to the back of your textbook in the appendix. I believe it's appendix page 5. That shows you all of the atoms that are attached to these molecules, so that you can clearly see that they are indeed 2 different molecules. This first molecule that's produced is what we call dihydroxyacetone phosphate. The second one that is produced is called glyceraldehyde 3-phosphate. Now, we have learned a little bit about enzymes in this chapter as well. Enzymes are very specific at their active sites for particular substrates. The next step that carries us into the energy capture stages of glycolysis will only recognize one of these 2 molecules. The only one that it's going to recognize is this glyceraldehyde 3-phosphate. So the cell has to do something about this dihydroxyacetone phosphate if it doesn't want to lose half of the energy associated with this glucose molecule. So what the cell does is it has an enzyme that in a separate step converts, it actually is what we call an isomerization reaction. So isomerization. And what the enzyme is going to do is it's going to convert this dihydroxyacetone phosphate simply by rearranging the atoms. It's going to turn it into a second molecule of glyceraldehyde 3-phosphate. So at the end of this step, before we enter into the energy capture phase of glycolysis, we're looking at now having 2 molecules of glyceraldehyde 3-phosphate. So in that last phase, we have to do everything twice. We have to perform each enzymatic step on each one of these glyceraldehyde 3-phosphates. Now, the names that I've listed, these are all important names that I want you to know for the test. You need to know that we start with glucose, that when we invest the ATP to phosphorylate the glucose to yield fructose 1,6-bisphosphate, this is the special molecule we get that helps us to weaken that middle bond so that we can lyse it into this dihydroxyacetone phosphate and glyceraldehyde 3-phosphate. Now, this dihydroxyacetone phosphate is going to be a major feed-in point when we start to break down lipids. We are going to feed in from a part of lipid catabolism right here at this point. So this is an important name to recognize. And of course, the glyceraldehyde 3-phosphate is also an important name because that's what's going to carry us into the next phase.